Hello everyone, and welcome to our invertebrate exploration lab. The entire lab today is about exploring the part of the phylogenetic tree where we see the broadest diversity of our invertebrates. So I'll start by hopefully recapping a bit of our lecture material, since y'all, and kind of shorthanding it as well. Of course, we all know that life arose in an aquatic environment. Bacteria, archaea, and the eukaryotes define our three major domains. But within the eukaryotes, we see a wide breadth of protist diversity. Of these different eukaryotic groups, one of them were the opisthecons, in which we see animals and fungi diversifying. Down the animal lineage, we see the things that derive their energy by heterotrophy, stuff that would eat other things by ingesting it. And we have some of our simplest animals as the first diverging lineages there. The poriferans, or our sponges, and the lineage with radial symmetry, the cnidarians, like your jellyfish. And then we come to the breakpoint that matters the most for us today. The breakpoint of the protostomes versus the deuterostomes. Now, the deuterostomes, like you and I, develop their mouth second. The protostomes develop their mouth first. And it is within this lineage that we see two broad breakdowns that lead us to the vast diversity of invertebrate life on planet Earth. With this primer, we're going to head over and take a look at some of the phylogenetic trees in the lab, because I think it's actually interesting. Over my time here at West Shore as an instructor, the phylogeny of these protostome lineages have actually changed. So you'll be looking at, the first thing that we'll be doing is looking at a tree from 2018 and see how it's been updated in 2021. See y'all over on my iPad.
All right, everyone, so here we are with our phylogeny from 2018. Now, for a bit of general topography here, you've got the sponges, the poriferans, as basal to the entire tree, with the cnidarians coming out after them. And then we, of course, get our differentiation between protostomes and deuterostomes. Now, we're not going to really focus on the deuterostome lineage here. That is, of course, where we see all of the vertebrates. We're going to focus instead on our protostomes. Now, we know that the lophotrochozoans and ectozoans are two differentiating branches, and in our greater discussion of the insects today, we're going to not focus so much on the lophotrochozoans. You notice that the vast majority of it is kind of collapsed into a polytomy, and for now, that won't really change, as you can see in our tree down here. You kind of have a massive polytomy. So that relationship doesn't differ, nor does it differ when we're looking at poriferans and cnidarians. What really changes between 2018 and 2021 is the relationships within the ectozoans, and in particular, these arthropods. So in diagramming these specific relationships, looking in the arthropods, we'll have one branch. Let's go ahead and make this a little bit thicker. We have the first branch that are our myriapods and our insects as sister to one another. And then on our other branch, we'll have crustaceans and the arachnids, or the chelicerates. So if we move this off of the tree, we see that the myriapods and the insects are sister to one another, and the crustaceans and chelicerates are sister to one another on independent branches. Now, if we look at the more recent tree, what do we see within our myriapods, crustaceans, and insects? Well, we see that the chelicerates actually act as our outgroup, that we have the myriapods after that. So, whoa, that's weird. We have the myriapods now more closely related to the chelicerates. And then within that, we have several different groups of our crustaceans. And then insects are lumped in within the crustaceans in something called pancrustacea. So the relationships as of 2021 are very different compared to what we see even just a few years ago. Though I will say that the phylogeny published by the Fort Worth Zoo was one of several different competing phylogenies, and this topography that it maintains today, or is more popular or more in vogue today, that's even the terminology that Wikipedia will use, has existed since 2005. There's been some variation as to where things are. Most notably, you'll notice that the trilobites, this now very popular but extinct group, if we're placing it on our tree currently, is here, sister, or just outside of the Chelicerata. So we have a very, very different topography where we kind of have a stepwise progression from the chelicerates to the myriapods to several different independently evolving lineages of crustaceans. There's been lots of different ways to evolve being a crab, which is pretty nuts to me. And then insects are actually a subset of these crustaceans. And I bring this tree up because it's kind of one that jives with a lot of what I've seen in terms of morphological evidence. There seems to be, at least it fits with the way that I've been taught biology. So, of course, make what you will of it, and perhaps some of you will go into phylogenetics, genetics, and systematics to try to better answer this question. But I'll try to keep on top of what's going on in the research world in terms of how these different groups are related to one another, in order to help better inform our understanding of the evolution of these species through time. Now, of course, a lot of these lineages are over 400 million years old. They have been around on planet Earth for an epic amount of time. And so it can be kind of hard to trace these branches that broke off from one another so long ago. So with that, I hope that y'all can go through and answer these questions in the lab. Of course, you should be able to discuss protostome and deuterostome development and compare relative diversity. Keep in mind that insects are incredibly diverse and insects are protostomes. So I don't particularly care how many different backboned organisms are on planet Earth. Not much compares to our insects. Please, again, discuss the topology of our different trees, and then take some time to think about which lineages, particularly in the arthropods, and of course the vertebrates, have made it onto land. So, I've actually excluded the vertebrate lineage here. Interesting. I will have to correct that in future years, but for now, c'est la vie. Maybe it's something you catch when you're doing the lab on your own. And then, of course, be able to discuss why spiders are not considered insects and how body plans have changed over time. I'll see y'all when we get to discussing our insect groups and a recapitulation of the giant phylogeny that y'all be building out of epoxy-filled samples.
All right, everyone. Now we've gotten to the meat of the lab today, because I don't want to just look at the invertebrate diversity and leave insects as insects. We know that insects are incredibly diverse, so we need to dive into the diversity of insects that we see, and that's the whole purpose of the lab today. And now I will have a lot of hands-on samples for y'all to be looking at. We have plenty of insects sealed in epoxy, some absolutely gorgeous samples, as well as some that might be a little bit more spooky or scary, depending on your fear of different things. And I want y'all to be looking at these, touching them, and then thinking about where in the phylogenetic tree all of these different species are. Because I am going to provide for you a phylogenetic tree for insects to the level of detail that I hope you'll be able to keep track of. We have silverfish, dragonflies, and damselflies, and then as one large clade, grasshoppers, crickets, stick insects, cockroaches, and termites. And then we have another branch that will have our true bugs, our beetles, ants, bees, and wasps, butterflies, moths, as sister to flies. Now, later on in the lab, y'all will be coming up with traits and examples to consider ways in which you can keep track of those relationships. But for now, given that tree and the tree that we had before, I'm going to give you guys a set of samples, and I want you to build the phylogenetic tree for that. So we have our samples A through P here. Going to be quite a nice phylogenetic tree. And now on video, I will go through in alphabetical order and kind of give you a little introduction to each of these different samples. So A is this rhino beetle, quite beautiful. Notice the amount of legs that it has. Our B is this moth with its different life stages. Sample C is this octopus with its 8 legs. Think about how that relates to our beetle that we showed you earlier. Here we have a jellyfish, Aurelia. Long story behind why that's one of my favorite names, but jellyfish are certainly important to this tree, and hopefully you can put them on the tree very quickly as they're there fairly early. Next up, we have a chitin, a shelled organism. Let's see if we can get the other side to roll over here. Maybe not. See, hold it upright. There we go. Something like that. And then we, of course, have a starfish. That will be very important for anchoring your species on the tree. And then next up, a round worm, which is an example of a nematode. And then we have a fully dissected earthworm. See all the different structures inside of an earthworm. I find them really cool, but you'll be placing the earthworm on the phylogenetic tree. Then we have a glass jar of creepy crawly millipedes. Hopefully they're far away on the tree from where I am. And then we have a cluster of organisms here. Brittle stars, sea urchins, sand dollars, sea cucumbers. Figure out what they are related to on this tree. Then of course we have one of my older brother's favorite organisms on the planet, snails, with their helical shapes. These gastropods are incredibly interesting, though I don't want to see them in my garden. And then not for the arachnophobic, we have a spider, sample L. Sample M being our crab. Sample N as our scorpion, quite spooky. Sample O, the opposite of spooky, quite beautiful, a beautiful butterfly. And sample P, a dragonfly. These carnivorous things that now are small but once had long wingspans. And so as indicated in the lab, y'all will be using these samples. You can abbreviate them as A through P or use their descriptions. I don't actually care if you remember that the jellyfish is sample D. You can just go ahead and put the jellyfish. But build a phylogenetic tree out of those samples and indicate some of their evolutionary relationships. Consider what groups are basal, what traits are shared, and I will see y'all after the coup de grace for this lab. All right, so how was that, everyone? Hopefully you had a little bit of fun. If you're in class, putting together the insect phylogeny, putting together the invertebrate phylogeny, moving around the different pieces, and seeing what kind of traits are similar or different, what traits are retained, what traits distinguish different lineages. That, of course, is going to play massively into your overall understanding of the invertebrate phylogeny. From there, we're going to move on to taking a look at the actual body plan of an insect. So here we have a pre-dissected insect for you. It has parts labeled 1 through 15. Of course, for some reason, 14 and 15 are not labeled. So 14 and 15 are not labeled here. But we do have the corresponding parts in this sheet. Now, I've, for your lab, alphabetized all the parts and asked you to maybe write out a quick definition of these, but most importantly, I want you to sketch out the different components of our insect. Because it's really interesting to note these cases of convergent evolution, where we even refer to the largest part of the limb of an insect as its femur, and the secondary part of its leg as its tibia, and then it has a tarsus. So we do see examples of convergent evolution in general body plans. It's really weird to think about that I have 3 parts to my forelimb, part 1, part 2, and part 3, and you have much the same in the legs of an insect. So go through and identify and label this sketch, just in case on the day of the exam, I give you a sketch that I've made, or perhaps a better artist, and I ask you to label a lot of those similar features. The next part of this lab is focusing a lot on examples of convergent evolution across the phylogenetic tree. So it's important to note when body plans, even when they're not shared by ancestry, end up fairly similar. Keep that in mind as you go through and identify the parts of your insect, and then continue on to give unique features about our different insect lineages, going from our silverfish to dragonflies to grasshoppers and stick insects to the cockroaches and termites that they're sister to the true bugs, beetles, and continuing on with ants and bees and ending up with butterflies and flies. Because ultimately, I'd like for this phylogenetic tree to stick in your head. Of course, continue on with some of these questions, reflecting on your own knowledge, plus reading further on Wikipedia. And I will see y'all for the last part of our lab, where we briefly go over the insect life cycle and see whether that has evolved in parallel in a couple different lineages. All right, so now we've arrived to the last part where we look at the insect life cycle. And for this, we happen to have a very neat little setup. So let's pan over and take a look at the 3 different types of insect metamorphosis. Gradual metamorphosis, where the insect will go from egg to a nymph stage that resembles the adult to just a larger version of the adult. In gradual metamorphosis, basically you're not getting a change in body form after the egg, it just gets bigger. In incomplete metamorphosis, you again start out as a tiny little egg, and your nymph stage vaguely resembles your adult stage, but you'll notice some very distinct differences, like these wings in the adult cicada versus the nymph cicada. it is an incomplete metamorphosis because they look similar, but not exactly the same, comparing it to our gradual metamorphosis where it's just getting bigger. Now, of course, the metamorphosis that you are most likely familiar with, whether you read Franz Kafka or are familiar with butterflies, is complete metamorphosis, where you begin as a tiny egg, progress into a larva, then you have a pupal stage where your body is literally broken down and reconstituted in order to form your adult that looks completely different from your larval stage. A complete metamorphosis because it is a complete body change. Now your task is to go back to our phylogenetic tree and think about where along different branches do we see gradual metamorphosis, incomplete metamorphosis, and complete metamorphosis? Do we have lineages where this is evolved convergently? Remember, this entire group, the ecdysoans, are all focused around molting their outsides. So metamorphosis is a fairly common process. In some cases, particularly like in other lineages that may not be insects, you may encounter more of a gradual metamorphosis. I am indeed looking at you, Mr. Krabs. So please, as a last revisiting of these topics, so hopefully I'm encouraging you to really understand the basics of the insect phylogeny, go back and take a look at their life cycles. And that'll do for this insect phylogenetics lab. Hopefully this has helped build on your understanding of insects. Hopefully it's made them a little bit less gross. They're an essential component of our ecosystems. And with climate change, we are seeing a decline of insects globally. That's not good because so many things require these insects for food. Much more, we also need them to break down things that we make. So they are essential, and even if they're a little bit gross, we do need to care for them and understand them. See y'all next week.